All right, this is our last lecture on allele frequency change. So we're going to work through the assumptions of the Hardy-Weinberg theorem and talk about when they're violated, how does this change allele frequencies, in other words, how is evolution occurring, and can this make populations go out of Hardy-Weinberg equilibrium. So the first assumption we'll discuss is why we have to assume larger infinite population size, and that's to discount genetic drift. So we can define genetic drift as chance events, and these can cause allele frequencies to randomly drift from generation to generation. And these chance events can play important roles in the microevolution of small populations. So here's our gene pool at generation 0, 60% A1 alleles, 40% A2. Generation 1 then 50-50, and then generation 2, 40% A1, 60% A2. Another way of looking at it is like this. So generation 1, P 0.8, Q 0.2, really different. Generation 2, same with generation 3. So the numbers drift from generation to generation. That's where the name genetic drift comes from. It's also an example of what we call sampling error. The fact that the smaller the sample, the greater the chance that the outcome will deviate from idealized results. So in a small population, the existing gene pool may not be accurately represented from the previous generation just due to sampling error, which individuals mate with which other individuals, these kinds of random events, or which individuals might get killed or something, or just not mate that season. So here is a population of only 4 individuals, and actually 8 populations. One of them is put in red so you can see. So on the y-axis we have the frequency of the A1 allele, and the x-axis the generation time. So notice by 20 generations, all populations, the alleles are either fixed, that means a frequency of 1 at the top of the graph there, along here, or extinct, a frequency of 0 along the bottom of the graph. And so it only took less than 20 generations for that to occur in a small population of size 4. If we increase our population size to 40, we see that it takes longer for alleles to become extinct or fixed, and in only one population is the allele fixed, and that happens at about 40 generations. And you've got 3 populations where the allele became extinct, and it took 40, more than 60, and more than 80 generations respectively. And then we've got about 4 populations where both alleles were still in existence after 100 generations. Now the red line is just there to show you what happens in one population, so it's easier to see. If we increase our population size to 400, after 100 generations, both alleles are still around, although there's been a lot of sort of warbling around from generation to generation, but no extinction or fixation. So genetic drift has a smaller effect on larger populations. Here's a neat experiment where they took fruit flies, and they had 107 populations, and they raised them for 19 generations. So at the top there, you can see the allele frequencies there are mostly clustered in the middle, right around 0.5. And then as time goes, you can see that the bars at the either end of this linear graph increase in size, indicating that in more populations, the alleles were becoming fixed or extinct. And you can see that there's fewer populations where both alleles are still present. And so this shows the effect of genetic drift over 19 generations in 107 different populations of fruit flies. This is heterozygosity loss due to genetic drift. So the y-axis is frequency of heterozygotes starting at 0.5, and then the x-axis is time in generations. So in a population of only 16, by 100 generations, there's almost no heterozygotes. A population of 64, there's about 10% heterozygotes after 200 generations, Population of 256, it's dropped by 10%, and then a population of over 1,000, it's only dropped by a couple of percent over 200 generations. So as you can see, genetic drift, again, has much less effect on larger populations. So we'll look at a couple of specialized examples of genetic drift. One's called the bottleneck effect of a wine bottle with a narrow neck. So this is a catastrophic decrease in population size caused by natural disasters like fires, floods, earthquakes, or by overhunting. And so it subjects the population to the effects of genetic drift or sampling error. So by chance, after the catastrophic event, certain alleles will be lost. Others may be more common than they originally were, and others alleles may be less common than they originally were. And the long-term consequence of a bottleneck is a drastic reduction in genetic variation. So when we see less genetic variation than normal in a population, we figure it's probably gone through a bottleneck. An example of that is with the northern elephant seal. In the 1890s, they were down to only 20 individuals of that species left. Then they became protected, and by about 100 years later, there was almost 130,000 of them. But examination of 24 genes showed no genetic variation. They were all fixed for a single allele. So these seals are still susceptible to extinction. A new disease or something could come in and wipe them out since they have such low levels of genetic variation. And here's a pair on the beach making more elephant seals. The male's on the right with the big blubbery nose, and then the one with the normal looking nose is the female. Now, second specialized example of genetic drift is the Foundry effect. And this is genetic drift in a small colonizing population. So they can affect populations colonizing remote islands or new lakes for an aquatic organism or new mountaintops. One seed or a single pregnant female would be the most extreme cases of founding of a new population. But example comes from an island in the South Atlantic called Tristan da Cunha. And in 1817, a Scottish family went down there to found a population. And just by chance, one of them was a carrier for a genetic disease called retinitis pigmentosa. So it's progressive blindness in the little a homozygotes. So as they age, they become blind. By the 1960s, there were 240 people living on Tristan da Cunha. 4 people had retinitis pigmentosa and 9 people were carriers. And so this is a much higher frequency of the retinitis pigmentosa allele on that island than anywhere else in the world, all just because by accident the population was founded with one individual that was a carrier for it. And so there's the location of the island, sort of halfway between South America and Africa, and then that's what it looks like. It's a volcanic island, I guess not connected to any other islands or anything. So summarizing genetic drift then, it's more pronounced in small populations, but it can still happen in large populations. Results in allele frequency change and loss of genetic variation. Either alleles become extinct or fixed if genetic drift is operating, and it can cause genetic divergence between populations as well. So moving on now to random mating. So why do we have to assume random mating? Because of what non-random mating does. And we'll discuss 2 types of sortative mating and inbreeding. We'll discuss them in that order. And non-random mating can come about by choice or by circumstance. It changes genotype frequencies, but it doesn't directly change allele frequencies, so non-random mating is not considered an evolutionary mechanism. So sortative mating, this is mate choice where individuals choose mates that are like themselves in a certain trait. In our species, it tends to be with height. So taller women prefer to mate with taller men. They say an average, in the average couple, the man's about 10% taller than the woman. Among snow geese, they come in 2 plumage color phases, white and blue, which is really gray. And anyway, they prefer to mate with others of their own plumage color. And so what this does is increases the homozygous genotypes and decreases heterozygotes for plumage color loci. So here are the 2 color morphs of the snow geese, Chen cerulescens, the white form, and the gray or blue form. And again, the white ones prefer to mate with the white ones, blue ones with the blue ones. Now on to inbreeding. I'm sure we all realize that that is mating with individuals that the organism is genetically related to, mating with your kin, in other words. The most extreme case, of course, would be self-fertilization, which plants and some invertebrates can do. It reduces heterozygotes while increasing homozygotes. And so phenotypically, more homozygous recessive little a individuals exist, and they are then can be selected against. So it originates in two ways. In some species, they choose to mate with relatives, and in other species, their populations are subdivided such that the only individuals around you to mate with are your relatives. We say that each person is said to have 4 lethal alleles which kill when paired. So that's probably why you don't want to have kids with your brother, because they're more likely to be stillborn. Now we scientists want to quantify things, so we could come up with a coefficient of inbreeding called F, and it's a probability that 2 alleles in an individual are identical by descent. It's computed from pedigrees, and you just multiply the probability of inheritance of that allele of interest in each generation. So this shows you alleles identical by state. And so at the bottom there, that individual, that the A1 homozygote gets the red A1 from one great-grandfather and the blue A1 from another great-grandfather. So this is alleles identical by state, not identical by descent. So this would have a F of probably 0. But alleles identical by descent, the A1 individual here at the bottom of the pedigree got both those A1 alleles from his great-grandmother, great-great-grandmother. And so this would be an example of inbreeding then. So this is an interesting one. Suffering reduces the proportion of heterozygotes by half in each generation, leading to a completely homozygous population. So about 6 generations of full sibling matings, we have an F of 1 and no heterozygotes at all. In number 3 there, matings between siblings increases the percentage of homozygotes relative to random mating. So we have an F coefficient of 0.25 after 15 generations of sibling matings, and we have a population that's about 98% homozygotes. Now, mating between first cousins also increases the F up to about 0.0625 after 15 generations, and we have about 68% or 2/3 approximately of the population is nothing but homozygotes after 15 generations of first cousin matings. So, inbreeding depression is defined as the what I'm trying to say here, exposes deleterious recessive alleles to the effects of selection. Genotype a appears more frequently and is usually selected against. This could be lethal, or it might just merely reduce the fitness of the individuals that are a derived from kin matings. So, looking at it in plants, biennial plants take 2 years to complete their life cycle. The first year they just grow a rosette of leaves, and then the second year they bolt or send up a flower stalk, producing flowers and seeds and reproducing. As we can see, these blue bars are much higher than the white bars. So in the second year, when they go to undergo sexual reproduction, this is when we see the effects of inbreeding really kick in, and the coefficient of inbreeding depression increase. And so they have fewer flowers and lighter seeds and so on and so forth. We can see in corn, as the inbreeding coefficient increases, our average yield of corn decreases from like 65 bushels an acre for no inbreeding, completely inbred population drop down to maybe 25 bushels per acre, and plus the corn cobs are going to be smaller with fewer seeds on them. Inbreeding also affects animals, of course. In Europe, there are little chickadee-like birds called tits, and in this one species called the great tit, you can see here as the inbreeding coefficient goes up, the number of eggs that fail to hatch in a clutch increase. So as we get to 1/4, 2 eggs per clutch fail to hatch, and these little birds probably only lay 3 to 4 eggs per clutch anyway. So we're looking at lots of reproductive failure in inbred populations of these birds. Also can affect humans. Here's some results from inbreeding in Japanese children. So mortality of children through age 12 years of age is quite low among unrelated people, becomes higher in second cousins, and even higher in children whose parents were first cousins. Another way to look at this is with this graph, where the y-axis is mortality rate among children of first cousins, the x-axis mortality rate among children of unrelated cousins, and the equivalent would be right along the line running up there. The line is equal mortality rates for both. And you can see almost all the red dots. Each red dot is a population, not a couple, but a population of people. And almost all the red dots fall on the left side, the upper side of that line that is equal mortality, indicating that in populations where there are first cousin matings, mortality rates are higher among the children. So that's all I've got concerning random mating. Let's talk about what mutations can do. So mutation rates are usually too low to change allele frequencies appreciably. So here we see it takes 1,000 generations for the mutation rate to change allele frequencies from 0.9 to 0.8. So mutation rate is usually too slow to make any kind of measurable difference. It's not going to knock populations out of Hardy-Weinberg equilibrium, is what that means. Mutation rate is given by the Greek letter mu, and for a mutation to be measured, it has to produce a distinctive phenotype. It's got to be fully expressed, completely penetrant, and it can't be mimicked. The phenotype can't be produced by drugs or chemicals, in other words. And normally, dominant alleles mutate to recessive ones, and that we can't detect, so we have to have a situation where a recessive allele mutates to a dominant allele, and then we can see a phenotypic change there, and we know that that individual is a mutant individual. And fortunately for our species, achondroplasia is a type of dwarfism where the torso is normal in size, but the head is larger than normal, and the limbs are much reduced in size, so that people have a short stature. And again, it's a dominant allele, so little d in regular people mutates to big D in achondroplastic dwarfs. And in 250,000 live births, they found the mutation rate to be 1.4 times 10 to the minus fifth, plus or minus 0.5 times 10 to the minus fifth. So before the mutation rate, P sub naught would be 0 and Q sub naught would be 1, because everyone's little d, normal. So the next generation, the frequency of the big D allele would equal P sub 1, which is just going to be Q sub 1 times the mutation rate. So in other words, and since Q sub 1 is 1, it's merely the mutation rate. So now if we have a mutation rate of 1 times 10 to the minus fifth, like for the achondroplasia, it's going to take 70,000 generations for P and Q to equal 0.5, okay? If the mutation rate proceeds that way and no other evolutionary mechanisms are at work here. So anyway, that's very, very slow. It's not going to change the allele frequencies enough to move a population out of Hardy-Weinberg equilibrium. This shows you that mutation rates can reach an equilibrium. So at the top here, we've got the forward mutation rate of the pale circles mutating to the dark ones and the reverse mutation under that. And as we get more dark G2 alleles accumulating in the population, the reverse mutation rate increases, and eventually an equilibrium where the two mutation rates are about equal is achieved. This is another way of looking at that. Number one there, P decreases owing to forward mutation, and 2, P increases owing to reverse mutation, and then at 3 at equilibrium, forward and reverse mutations are equivalent or balanced. So now let's look at gene flow, how that can change populations. So remember, gene flow is migration, and we can calculate the effect of this using this cool little formula where delta P is the change in the allele frequency of P in one generation, and P sub M is the frequency of the dominant allele in the immigrants, and M is the coefficient of migration. That's just the proportion of individuals migrating into the population, like 0.1 for 10%, 0.5 for 50%, something like that. So throwing some numbers in here. So P is 0.4, 10% of the population is migrating in, so that's 0.1, and then P from the immigrants is at 0.6. So plugging the numbers in that equation, we get 0.02. So from this, we see that allele frequencies have changed from 0.4 to 0.42 in just a single generation with those other parameters. So gene flow could knock populations out of Hardy-Weinberg equilibrium. And so this shows you how migration or gene flow can work on our little imaginary mouse populations. Our initial allele frequencies, everybody's A1, no A2. Then there's our gene pool, and then the little round circles are our zygotes, and then our baby mice. And we have 200 A2 individuals, so blue mice coming into the population. And now we can see our genotype are different there, 800 A1, 200 A2, and now our allele frequencies are different, gone from 1 and 0 to 0.8 and 0.2. So this shows how gene flow or migration can alter allele frequencies. And this is another way of showing that, how migration from population 1 into population 2 changes allele frequencies. One example in our species is that in Eastern Europe, there's an 18% excess of type B blood relative to Western Europe. And type B blood is much more common in Asia than in Europe. And this is due to gene flow from Asia or Mongolia. Think centuries ago, good old Genghis Khan and his Mongol hordes came across Asia and made it about halfway through Europe. And so this map shows you, as you go further east in Eurasia, the frequency of type B blood increases from 5% or less up to about 30% when you're over in Western Asia there. Now there's something also called the Wallin effect, and this is a decrease in heterozygotes, excuse me, and then an increase in the homozygotes from what's expected based upon allele frequency. So in other words, there's going to be less than 2pq heterozygotes and more than p squared and q squared homozygotes. And we often see this in highly subdivided populations that have inbreeding in little groups called demes. So it really results from two factors: population admixture. If we sample a population and it was really two populations that just merged and we sampled them before they had a chance to undergo any random mating, we might see the Wallin effect. Or secondly, if we take a highly subdivided population and treat it as if it doesn't have any subdivisions, so that's called a panmictic population, then we're combining individuals from different demes, and then we'll see the Wallin effect as well. So the second one would be kind of like a mistake by the researcher, or just not knowing that your population is highly subdivided. So you can sort of see it like this. At the top, we've got population 1, where our allele frequencies are 0.5, population 2, where they're 0.8 and 0.2. These populations merge, and you sample them before they can mate randomly. We see that the Hardy-Weinberg predictions would be 0.455 heterozygotes, but in reality, it's only 0.41 heterozygotes. And you can see for both homozygotes, it's higher than the Hardy-Weinberg prediction. And that's the Wallin effect. So again, Hardy-Weinberg says we should have 45.5% heterozygotes. In reality, we've only got 41% heterozygotes. So view it's kind of like this: population A and B merge. We sample them because what we're doing is sampling two populations at once. We just don't know it. But after, again, one generation of random mating, Hardy-Weinberg equilibrium would be restored. Remember, that's the third prediction of the theorem. Now, lastly, let's talk about selection. So here we're going to combine natural and sexual selection. So mostly we'll talk about natural selection that was first discovered by the Englishman Charles Darwin. And he wrote about it in his book, The Origin of Species, which came out in 1859, and says that the environment favors certain phenotypes, hence their genotypes. So therefore they have a reproductive advantage and their genes will accumulate in the population over generations. And then adaptation is the end result of evolution by natural selection. So the theory of natural selection has some postulates. Individuals within a population vary. That's due to, of course, genetic variation. Some of these variations then are genetics, and so they're passed on to the offspring. And in every generation, more offspring are produced than can survive. Darwin called this the struggle for existence. And then lastly, survival and reproduction are not random, but those individuals that reproduce the most have the most favorable variations. In other words, those individuals that have higher fitness. So fitness then is a measure of survivorship and reproduction of individual organisms. But again, survival without reproduction is still a fitness of 0. So it's your offspring that carry copies of your genes. So to be evolutionarily successful, you need to pass on those genes, have babies. So an evolution occurs when natural selection causes changes in the relative frequencies of alleles in the gene pool. Remember what the gene pool is and what alleles are. There's a picture of Darwin late in his life. And Alfred Russel Wallace, a younger Englishman, also discovered natural selection independently, although this was much later than Darwin. So Darwin's given more of the credit than Wallace. So here shows phenotypic variations, population of snails. Look at all the different color patterns present. And so nature could edit out some of these if natural selection was working on this population. We're going to look at an example called industrial melanism. So again, industrial melanism is just the name of this example of evolution by natural selection. And it's in the species of moth that lives in England, where there's a color polymorphism. A dominant allele, big C, caused the moths to be melanistic or very dark in color, dark brown to almost black, and a recessive allele, little c, causing them to be pale, like a pale greenish gray color. So prior to the Industrial Revolution, of course, before 1850, the air was not polluted. And the trees over there were covered with lichens. Lichens are most lichens are pretty sensitive to air quality. And so at that point in time, 99% of the moths were the little c genotype, were pale in color. Because remember, moths are nocturnal, so they spend the day sleeping on the tree trunks. And those that can blend in are not eaten by the birds that feed on them. The birds are vision-oriented predators, so if they can't see the moth, chances are it'll survive to live another night and reproduce. So pale moths are cryptic, like we see here. The original situation was at the top, where you can see the pale moth. It's very hard to see by the lower arrow there in the top panel. The dark moth really stands out. But you see in the lower panel, the environment changed, and then the dark moths became more cryptic, and therefore their fitness went up, and the pale moths became less cryptic, and their fitness went down. So in the late 1800s, of course, with the Industrial Revolution, lots of air pollution was generated, and that killed the lichens on the trees. So now the melanistic moths that were big C or big C little c were their genotypes that increased in fitness. And because they're more cryptic on these dark tree trunks that don't have lichens on them. And so this dark morph of the moth became the most abundant morph in 50 generations or less. By the 1960s, the Brits got their act together and passed environmental laws cleaning up the air. And we returned to the original situation where lichens recolonized the trees. And now the moths have higher fitness. And in Liverpool, England, the dark moths went from more than 90% of the population to just over 50% of the population in 26 years, from 1959 to 1985. So we say genetic variation is the raw material for evolution by natural selection, and that genetic variation happened to be that there were 2 alleles in the population for color pattern, big C and little c. If 100% of the moths would have been pale, then they would have probably just gone extinct, all gobbled up by the birds. And the selection pressure then, the reason that it mattered what color the moths were, was bird predation. So we can incorporate selection into our mathematical model too. where S is a selection coefficient selecting against the little a allele and its maximum of 1. So if S equals 1, then little a genotype is lethal. If S equals 0.01, that means then that 99% of the little a individuals can reproduce. So we're only slightly selecting against the little a allele. But selection takes a long time to get rid of a little a allele, to make it go completely to 0, because it can hide out in heterozygotes. So there'll be a few big A, little a individuals in the population. We call this a heterozygote refuge. So little a will decrease quickly at first and then maintain a low frequency presence for many generations. As we can see here, in 6 generations, it can drop from 0.5 to 0.125, 50% down to 12.5%, in other words, but it can hang out at a frequency of about 1% for 70 to 100 generations, again, in that heterozygote refuge. So that's what this graph shows at the top there. We're starting at 0.5, selecting against little a, and then for 200 generations, it's still there, albeit at a low frequency. Now, if we want to select for it, we can bring it to fixation in about 40 generations, as the lower graph shows you. So that means by generation 45 or so, everybody's little a, no more big A left in the population. So then if our selection coefficient is at its maximum, we can calculate the allele frequencies in the future generations with this formula, where n is the number of generations since p sub naught and q sub naught, the starting point in time, if you will. But if S is less than the maximum, less than 1, by the way, S is from 0 to 1, then we use a different formula where the original frequency of Q minus S times Q squared over 1 minus S times Q squared is what the frequency of Q will be after a single generation. So Q sub 1 just means 1 generation later. So this shows our evolutionary mechanisms. We've got our gene pool there, drift acting on that, then selection acting against the blue zygotes. Then we've got gene flow migration coming in, and then of course mutations working as well. So that's it. Good luck. And we are done.